Thank you very much, Anna. It's a pleasure to be here. Thank you for the invitation to speak. I was invited to this meeting last year, but ended up flat on my back in a hotel room at the time. So I'm grateful that you persevered with me and invited me back again. I'm a GI oncologist, so I treat people with gastrointestinal cancer. So I apologise in advance that many of the nuances and the details of management of this disease I won't be fully on top of compared to the many experts in the room. Okay, but I wanted to, and this is completely different to what you've been talking about, but just give you a sense of a project that I hope will continue to grow and expand, and is funded by the three organisations down the bottom there that you can see. So Ovarian Cancer Australia, the Australian Society of Gynae-Oncologists, and the CAS Foundation. Just by way of disclosures, the registry program, so we have quality registries in a number of areas, is funded by a number of pharmaceutical companies, although to varying extents, and they include AstraZeneca, who's the sponsor for today. So clinical quality registries, and these, remember, registries mean different things to different people. Population registries, such as the cancer councils have, is a different type of registry. A database on a clinician's computer is a different type of registry. Registries mean different things to different people, but the quality registries that I'm talking about are defined by the Australian Commission of Safety and Quality in Healthcare. They've been endorsed by RMAC, and they represent a framework to measure quality of care. Essentially, the principle, I guess, there's a lot of text on these slides, but just to say that the principle here is if you want to improve quality of care, the first thing you've got to do is measure it. If you can't measure it, you can't improve it, because you don't know what you're improving. And these are frameworks that are designed to actually measure quality of care, and in the process of measuring it, have got demonstrated capacity to actually improve quality of care. So they collect uniform data against a range of indicators, and then provide risk-adjusted and benchmark data back to clinicians. I'll give you an example of that shortly. So how they work, the first thing you do is you get a group of experts together, and that's challenging both for lots of reasons, but you bring a group of experts together and you ask them a very simple question. It's actually not that simple, but because often there's no evidence to conclude what's best practice. But you ask a group of experts, what is best practice? What is best practice in diagnosis? What is best practice in treatment? What is best practice in surveillance? What is best practice for the patient? What is best practice to treat this condition? And when you have an agreement on best practice, then you work out the indicator that measures compliance with best practice. So if everybody needs a biopsy, if that's the agreed best practice indicator, then the indicator is straightforward. How many women have got a diagnosis of have had a biopsy? So the best practice part of it's hard. The indicator is relatively straightforward. And then what you do is you collect the data, and that's a challenge we'll come to in a moment. You collect the data and you risk adjust. So the management of a 90-year-old is not the same as the management of a 20-year-old. Someone who's got very severe obstructive airways disease is not going to perhaps not have surgery for good reason. So you have to be able to risk adjust. And this is largely where administrative data falls down. Administrative data doesn't allow you to, firstly, it's designed for administrative purposes. And data designed for one purpose is often not that helpful for other purposes. Not to say that it can't be used, not to say that it can't be linked, but nevertheless limited by the very reason it's set up. So you risk adjust and then provide benchmark data back to individuals. So this is identified patients to identified clinicians to identified institutions, and showing them their respective data and looking at variation, because there's always variation, and when there is unwarranted variation, asking the question why. So variation may be quite appropriate, and patients are all different. But the question you're asking is when you see systematic variation in processes, in outcomes, what is the basis for that variation? And that in itself leads to improvements. So, as I said, a good clinical quality registry is very powerful in detecting unwarranted variation in care and acts as an early warning system for problems in care. But more importantly, I think offers great opportunities for research, and I'll mention some of those in a moment. The whole purpose of a quality registry is to collect a minimal data set, and when you get large data sets, you get difficulties in routinely collecting it and keeping that data set up to date. But it essentially is a cohort study, if you like. It's a long-term observational study which collects individually identifiable data that are uniform, minimal, and epidemiologically sound. And then, of course, allows you to link outcomes to processes of care. So one of the questions you often get asked is, where does a clinical quality registry and a clinical trial, and if you like, a hospital audit, how do they differ? And if we just look at the two on the left, the clinical quality registry, a CQR and a clinical trial, and just contrast them for a moment because they're different, but they complement each other. So a quality registry is for a whole population, whereas a clinical trial is usually a highly selected population. The funding comes from a variety of sources, as you might expect. The time frame's ongoing, whereas a clinical trial tends to start and stop, as you would expect, as it reaches certain defined endpoints. The purpose, and this is an important point, the purpose of the registry is to measure whether good care is being delivered. The purpose of the trial is to define what good care is. So what is good care? Is it an immuno-oncology drug? If that's good care, then is it being delivered? And if it's not being delivered, why isn't it being delivered? So the trial's important, the registry's important, and they complement each other. And to show you this, I don't know how well you can read this, looks like you can't. It's great from up here, but so this is a model of how these fit together. And I chair ACTA, the Australian Clinical Trials Alliance, and we have this model of what we're calling a self-improving health system. And it starts, is there a pointer? No. It starts with this idea of clinical quality registries, which is, they're all in the same colour, so sort of purplish pink. But you start with clinical quality registry and you find variation. And when there's variation, that may be due to a number of causes, but one of the reasons why there may be variation is there is uncertainty about the evidence base. So one group does X, another group does Y, not because any of them are wrong, there is uncertainty about what the right thing to do is. So the quality registry allows you to determine that variation exists. The trial process says, let's compare treatment A and treatment B since no one knows what to do. Let's compare test A to test B. No one's sure which is better. And then it comes up with a result. And then when that result is implemented into practice, the registry continues asking, is it being implemented? Is it being used? Where is there variation? Where does that generate hypotheses of further research? So in terms of developing a quality registry for ovarian cancer, certainly there's lots of anecdotal evidence of variation in care and variation in outcomes. You'll know better than me that survival's still relatively poor and hasn't changed dramatically over recent years. And as I say, the purpose of the registry is to at least say that insofar as the evidence currently exists, is it being used appropriately? Is the evidence base appropriately implemented in order to say that, yes, we need new treatments, and yes, the registry is not going to shift that curve up to 100%. But the registry will ensure that at least the evidence we have is being used so that when the next treatment comes on, it builds on the base of what we currently know is best care. I just want to draw your attention to the bottom half of this slide, which is about a prostate cancer registry. And I presume not many people have got up here and talked about prostate cancer too often, but I just want to illustrate an issue here. So here's a registry in prostate cancer, and one of the things that they looked at was surgical margins. So you have the prostate removed, and the tumour should be out and the margin should be clear. And they looked at positive margin rates in a group of men having a prostate surgery operation. And they looked at this across Victoria and found that one hospital had high volumes and yet they had a very high positive surgical margin rate, 2 standard deviations above the mean. So the surgical margin rate was excessive. So that hospital was an academic hospital. They did morbidity and mortality meetings. They did safety meetings. They did quality assurance. They had no way of knowing that they were 2 standard deviations above the mean until we could provide them risk-adjusted benchmark data. And then they knew. And what did they say? The data's wrong. Clinicians always say the data is wrong. So that's okay. It's appropriate to make sure the data's correct. So the data's corrected, and the results are the same. So then they say, well, the risk adjustment's wrong. So, well, how else would you like to risk adjust? So they risk adjusted on, how well differentiated the tumour was, poorly differentiated. And admittedly, you can't risk adjust against all the variables, many of which you don't even know what the variables are. They went back and said, okay, the data's correct. What are we doing differently? Why are our results different to the hospital down the road? And it turned out they had registrars or fellows doing surgery unsupervised. They fixed it, and their margin rates improved. It changed practice, and it changed practice in a way that they didn't mean to have positive margin rates. They didn't intend to have that. They didn't know they had it. And it's the power of being able to provide whole population quality data about quality of care. There's another example about prostate cancer, but rather than belabor it, it may come up in questions. The types of indicators that you collect, you can collect process indicators, structural indicators. I won't go through these in detail, and of course outcome indicators. So the range of clinical indicators that can be collected, and in the end, this is worked out by a process of sitting down with experts, and I include consumers in that discussion around experts, and actually figure out what are the things that measure best practice. These are some of the indicators that we are collecting in the current pilot. Again, I suspect too small to read for many of you, but they're the sort of things that you would expect when a group of experts sit down and say, what's best practice? And they come up with a range of measures of best practice. There's a number of indicators. This is a pilot. There are a number of indicators that aren't there and that could be built in, things like molecular testing. We've got referral to a familial cancer clinic, but not molecular testing results per se. Fertility preservation, is that being used in everyone in whom it's appropriate? Mortality within 30 days of chemotherapy. There are a range of indicators that you could add to the list. We have a starting point. We certainly haven't finished. And then we have to collect a range of measures that allow you to risk adjust. So, of course, you need to know about tumour morphology and tumours and stage, and you need to know about BMI. And there's a range of things that are required to be collected in order that you can actually do risk adjustment in some reasonable and consistent way. One of the things that I think is really important in the quality registry, and part of the reason we have identified patient data is to collect patient-reported outcome measures. I think that this is a real opportunity for us to actually to build that into this and to understand again where different interventions do or don't influence patient-reported outcome measures. Michael, I suspect, has reported at this meeting I think it's called the MOST study, where they've talked about the issues of symptom burden in people having chemotherapy, and of course this could be extended across the board in terms of the opportunities to understand what's happening at the patient level. We thought that we had limited funding for this pilot registry, and so we thought we'd do this by collecting data from existing databases. I mean, a registry, remember, doesn't create any data. It uses data that already exists, collects it, and then allows benchmarking to occur. So we thought we'd collect it from existing databases, and we were told that a number of surgical units or units around Australia had such databases, and so we thought this would be a test at least of an alternative mechanism of collecting the data. There's a fair amount of work involved in doing that because what we have to align definitions. You have to make sure that when someone else is talking about a debulking surgery, that we're talking about debulking surgery, we're talking about the same thing. So there's a fair amount of work involved, but actually the hard part of it is still getting the data out of those databases, and we're still challenged with that. But the way this works is that the units are actively, the units are seeing patients. They let us know, if you like, in this case Monash, but they let us know that a patient has got a diagnosis. They're screened and they're invited to participate in the registry using an opt-out consent model. This has been approved by ethics. It's done under an ethics and governance model, and that brings its own challenges. But nevertheless, the system, once you get the data, it's relatively straightforward, and very few patients opt out of this. One of the things that's very important about a clinical quality registry is the clinician engagement. And I used the example of the prostate example before of people having surgeons with looking at their prostatectomy data. The key issue here is that we are providing benchmark data to the clinicians involved, and this peer review process, sitting down with the next clinician and comparing results, is an extremely powerful way to actually present results and an extremely powerful way to induce change. And so clinician engagement is really critical to this, and that's why it's different, again, from administrative data. I used to have a role at Peter Mac as Chief Medical Officer, and we looked at administrative data all the time. And I can tell you that you want to see someone's eyes glaze over, you show clinician administrative data. You can see it, the glazing, it's in front of you. Administrative data is collected for administrative purposes. It's important for that purpose. It's not clinical level data. It doesn't, of its own account and independently, result in change in practice. So we currently, this is a pilot. We've got, it commenced about a year ago. We're recruiting patients around several states around Australia and from 10 hospitals. And we've got a number of participants at this point that are enrolled in the registry. Obviously early days, and the history of these registries that they are slow. The prostate registry took a number of years to get to the point where it now collects data on every man with prostate cancer in Australia on regular intervals. I mentioned that it was approved by ethics, and so now, and the ethics approval actually allows us to collect data on patients with other subtypes of ovarian cancer as well as other types of gynae-oncology cancers. The challenges, so I mentioned at a meeting earlier this morning that if you wanted to design a system that would slow down collection of data and research, that we've got the perfect system in Australia. Not sure that's something we're proud of. But nevertheless, we are progressing through this. We do have governance and ethics approval at a number of sites, and the data collection has started. I think this is my second last slide, and just to say that what's happening in terms of the future. So the federal government is developing a national policy about quality registries. The Medical Research Future Fund strategy document identified quality registries as a strategy that they wanted to support. There will be, I have no doubt, the cost of registries is largely in the data collection, and the future will no doubt bring automated data collection. We're not there yet, but it will happen. And that includes all the levels of sophisticated natural language recognition, uploads directly from pathology departments and so forth. So I see a really exciting future in relation to the access to data. There's been a few studies of randomised trials within registries, and it doesn't work for every trial. But it used to be thought, oh, that's a great idea, because actually what we're collecting the outcomes, so all we need to do is do the intervention, and it'll be a cheap way of doing trials. Actually, it's not cheap, but because the registry pays for the observation, so that changes the cost structure. But the beauty of a quality registry and doing a trial in a registry, and the power of it, and the reason why I would hope ANZCOG and other groups might think about this, is that these are trials in real-world populations. These are not trials in highly selected people with relatively indolent disease. These are real people having real disease and an opportunity to randomise them, whether it's about a behavioural intervention, a supportive care intervention, a monitoring intervention, a surveillance intervention, or indeed subsequent treatment interventions. Down the track, we'll be reporting these data to the public, and that's going to be challenging. It's happening in the US, it's happening in the UK, but it will happen in oncology. What we're trying to do is say to governments who want access to this data, and the public who rightfully say this is taxpayers' money, we want access to these data, what we're saying is let's get the data to a point of maturity where we can all believe it, and then the data will speak for itself. So I think that's another part of the future, which I guess is not so much threatening, but leaves people with a feeling of uncertainty as to how that's going to play out. But one thing you can be sure of, when you get public reporting of quality data, practices change. They change quickly. They change very quickly. And they will change for us as a community when that happens. But we need to get to the point where we have enough data and enough quality data to actually report. I'd like to conclude by thanking all the people involved in the work. I mentioned OCA, Ovarian Cancer Australia, so thank you, and the Cass Foundation and ASGO. We have a steering committee of people that have spent time. We have a number of clinicians and database managers. And finally, Natalie Herriot, who's done all the work here and sitting in the back of the room. So thank you very much for your attention.